I feel immensely honored to have been asked here to give this plenary talk at the Phys-soc, which is a word I've heard often from my friends across the pond, but have never actually been to before. So this is a thrill. I'm gonna tell a story, as Richard has intimated. In Richard's introduction, he pointed out that I like to work on weird things, and I have found that as my career has progressed and I have been entering further into my dotage, I seem to work on increasingly weird things. And this story I'm going to tell you about today is probably the weirdest channel I've ever worked on, and I want to indicate some aspects of the weirdness. But I also want to say that well, I'm not a physiologist, you know. I'm here at the Phys-soc, but I'm not a physiologist. I am always afraid of the complexities that cells, systems, organs, and so on present, and I tend to just work on single molecules. But in working on this story I'm going to talk about now, I have come to realize that there is a whole unseen and unimagined strange physiology that is associated with the ion channel I'm going to talk about. And though I don't have much in the way of physiological data, I have a little bit, and I'll show you. I will try to indicate places where I think there are interesting questions that perhaps young physiologists looking for strange niches to work in, where there may be very unusual things that will come up, should maybe consider going to. So for the last millennia, or whatever it is, the literature on membrane biology has been filled with a sort of a teeming zoo of monatomic ions, papers about the transport of monatomic ions in biology for all sorts of different physiological purposes. But there's been, over all that time, one such ion, obvious one in the periodic table, that's been left out of this carnival, and that is the fluoride ion, which until 3-4 years ago was widely considered to be simply one of these biologically irrelevant ions. And that idea was overturned by a breakthrough paper from the lab of Ron Breaker at Yale, who has discovered a series of fluoride ion exporters that live in microorganisms. And I'm going to talk about one of these. So here is the ancient challenge that my title refers to. That is, fluoride is everywhere in the aquea, in all waters on the planet, to the tune of something like 10 to 100 micromolar, and it has been there in the aqueous environment since the beginning of evolutionary time. Like other salts, it's just extracted from the rocks and so on, and it is to be found in not only toothpaste, but rivers and lakes and drinking fountains and the ocean to that tune. And in fact, this other indication here, I'll come back to a little bit later, is that it is found in quite a few plants that extract fluoride from the soil and deposit it in their leaves. The tea plant, which is shown here, is one of the most active fluoride accumulators for reasons nobody knows. And so it's all around the whole planet. So what's the problem? The problem is this. Now, this is my sort of rather naive view of life. And you need sort of two things to make the cell work. And you need food and sex for two particular purposes. You need energy, and you have to replicate. And, well, you know, this is probably also the male graduate student's view of life as well. But the point in this lecture is that both of those fundamental pathways are inhibited by the fluoride ion at environmental concentrations, that is in the tens of micromolar range, because these involve phosphoryl transfer enzymes that fluoride can kind of mimic a flying phosphate in the mechanism. And so bugs can't just mutate around this. This is something that organisms have had to deal with from the beginning of organisms. So it turns out, let me go into this in a little more detail. Why is it toxic to microorganisms, fluoride ion? It's a little more subtle than simply saying, well, you've got 50 micromolar fluoride there, and that would inhibit enolase or pyrophosphatase, and so you could inhibit nucleic acid synthesis. It's a little more subtle in that it has to do with the fact that microorganisms will sometimes float into acidic niches. That is unavoidable. And it turns out that fluoride has an unusual property, is that it loves to hold on to protons. You know, the pKa of HCl is around -6 or -7, depending on how you measure it. But the pKa, the acid dissociation constant of hydrofluoric acid, is 3.5. It's only 1 unit more acidic than acetic acid. So it's a weak acid. And what that means is that the famous, I think, to all physiology, beginning physiology courses, the weak acid accumulation effect will happen if a microorganism floats into an acidic niche, let's say at pH 5 here, in the presence of some fluoride. What you'll see is that you'll have a significant amount of HF in that medium, maybe a micromolar or two, which is now permeant to the membrane. HF goes right through lipid bilayers. So it'll get inside of the cell, but then it'll dissociate because it sees a regulated neutral pH inside. And what that will do is it'll accumulate. If there is no transport system for that anion, it will accumulate fluoride to the tune of the proton gradient. So if there's a 100-fold proton gradient, you're going to get a 100-fold accumulation of F-, and that will be really bad for the organism. So how can you undermine that? Is simply to put some sort of transport system, a pump, a channel, I don't care, in there so that the fluoride anion can permeate. And then the fluoride anion can follow the Nernst potential. And often microorganisms have rather high negative internal membrane potentials, and so this will keep, just even a simple channel would protect against such an insult from the environment. So this is why I think that the channel I'm going to talk about is found in many such microorganisms. I'll tell you a little bit of the breakthrough that Ron Breaker made. And this is 4 years ago. Ron published a paper in which he found, he's an expert RNA riboswitch and bioinformatician, and he found that many bacterial genomes have a structure in their RNA, their messenger RNA, called a riboswitch, which specifically binds the fluoride anion. And these control a gang of genes downstream from there. It upregulates a bunch of genes. And often in many bacterial genomes, Ron found that enolase and pyrophosphatase, these very same fluoride-sensitive genes, were controlled by this riboswitch. So when fluoride goes up in the cytoplasm for whatever reason, these things get upregulated to try to overcome the challenge. But he also found that there was a strange unknown function gene that often came along with these. It's a small, presumed membrane protein of unknown function that rode along in this gang of control genes, and he postulated maybe this is a pump, an export pump or something like that for fluoride anion, so as to, it would sort of make physiological sense. And that's when I got into this, because Ron gave me a call and asked some help for seeing about reconstituting at a protein level, because he couldn't get the paper published because the reviewer said you needed protein data, even though his genetic data was impeccable. In any case, that's how I got into this about 4 years ago, and it's been a delightful ride ever since. So we call this thing that I'm going to talk about Fluc. It was a fluke that I got into it, but it's for the fluoride channel. So now I have to just for a moment just say, I'm not going to talk about something that is there and that Ron also discovered, that actually he found in this same way that there were two kinds of presumed fluoride exporters that rode along in these genomes. Some bugs would have one, some bugs would have the other, and the other was a class of anion transporters from the CLC superfamily, which is something, as Richard said, I've been working on for a long time. That's why Ron telephoned me. And it just turns out some bugs have CLC fluoride proton antiporters. They're energy-consuming export pumps. And some have this thing, this Fluc thing. And I'm not going to be talking about the CLCs. I'm going to just be talking about the Flucs. The Flucs are found in many microorganisms, bacteria, yeast, various protozoa. They're found in primitive animals like tunicates and sponges, and they're found in many green plants, including Arabidopsis. But they are not found in any vertebrates. And this is also delightful to me, because it means that this project that I'm enjoying so much is entirely untainted by any relevance to human health. And so I don't have to deal with that nonsense. I want to just show you just one kind of set of experiments showing you Fluc in action in E. coli. E. coli has a single Fluc gene. And so what you can do is a very simple experiment where you just grow, so you look at growth curves of E. coli at pH 7 in wild type at different concentrations of fluoride in the medium. And you can see that wild type really doesn't care. But if you knock out that Fluc gene, well, you know, at 50 micromolar, which would be sort of environmental level, it doesn't care, but at higher concentration, it stops E. coli. But now as you go to lower and lower pH, you see, this is what I was saying about the weak acid accumulation effect, this hypersensitivity to fluoride becomes severe. And we can ask, is that because of this accumulation effect? That is, in the knockouts, is that because fluoride is actually being accumulated in the bug, in the cytoplasm? Well, that's easy to do. You just do this experiment and measure how much fluoride is inside the bug. And it turns out it agrees absolutely quantitatively with this very simple idea that if you don't have a fluoride transport system, you will accumulate fluoride to the same ratio as the proton inside and outside at different pHs. And so that is the line that you'd predict before you ever do the experiment, and that's the data, and it says, yeah, this is a classic weak acid accumulation effect here. So I think that's what the Fluc channel is doing, just protecting bugs from the combination of environmental fluoride and the inevitable low pH systems that they'll eventually find. I have no idea what it's doing in tunicates, but maybe someone would like to look into that someday. But don't expect much funding for that. I do want to just mention here again, because I like odd physiologies, and I also like plant physiology a lot. I think it's a health-related subject because, you know, if you're starving, it really doesn't matter if you have Alzheimer's disease or cancer. It really doesn't. Anyway, it turns out the tea plant is, as I said, one of the most robust fluoride accumulators from the soil known. Spinach also does it, some other things. And this is dark territory. No one has a clue why these plants do this. And I think it would be a pretty interesting little bit of investigation to go after. Someone, young person out there, starting your career. Well, maybe if you take me up on it, I'm ruining your career by suggesting this. But anyway, it appeals to me. And the other thing about this, about fluoride, is if you go and Google fluoride toxicity, you will be transported immediately into web internet crazy land. And I'm just, you know, there'll be the communist conspiracies to poison our precious bodily fluids from fluoridation, and there will be things like Dr. Mercola's Natural Health website that will tell all the Brits in the audience not to drink tea because it will certainly kill you because it is such a toxic heavy metal. That's Dr. Mercola's chemistry expertise comes to the fore there. Anyway, let's get back to this protein. So what is this protein? I'm just going to tell you, sort of going over the last 3 years of work in the lab. What is this? We come up with this protein, we can purify it, we can overexpress it, but what is it? We want to investigate it, but we don't know really, is it even, in what class of fluoride transporter is it? Is it a transporter? Is it a channel? And I'm going to show you that it is a channel, but I'm going to show you how we kind of crept up on that. So just to remind you, there's a fundamental mechanistic difference between channels and transporters. Channels move their substrates, their ions there, the cows going through the gate, in a diffusive, thermodynamically diffusive, dissipative way, and that tends to be fast. Channels tend to allow something like millions of cows per second going through the pore, okay? Transporters are very different in their mechanism. There's a cycle of conformational changes where each substrate has to be handled specifically by the protein, and of course, or you know, you can have rocking bananas or elevators or whatever you have, but I think this kind of shows it very well, and those are conformational, and they can often be energy coupled, and they are slow. So typically, you know, 100 cows per second maybe go through a typical transporter. So that's a very different sort of scale of unitary transport rates. And that motivates the first experiments I'm going to show you of trying to just see in an indication. Are we talking about something that looks more like a transporter or looks more like a pumpy sort of thing, pumpy or a channel sort of thing? We have two of these homologs we study. They're both bacterial, and we can reconstitute them into liposomes, and this is the assay. It's kind of an all or none assay. It's not that quantitative, but it just says, is there a channel there or is there not a channel there? Okay, and we'll come back to that. I'm going to need that a little later for another point later in the talk. So the way we do is we make liposomes and we reconstitute the purified protein in such a way that the liposomes, the reconstituted proteoliposomes, have only one copy on average of the protein. And so whatever rate we see is going to reflect unitary transport. Are we talking about 1,000,000 ions per second or 100 ions per second? And then, so we load it with high concentration of KF, that's the dark blue, and then we dump it into a solution that has a low concentration, that's the light blue. And we do this in ways that where most of the liposomes don't have any protein, but the ones that do have protein, because you're just putting a little bit of protein in there, have only one copy, and every once in a while there's statistically, you know, maybe 2 or 3, and we forget about that. Because we don't do experiments in my lab to more than plus or minus 20% anyway. So now Of course, the dark blue fluoride would love to come out of that down the gradient, but of course it can't do that because, at least in Massachusetts, we do not violate laws of electroneutrality. And so to get the fluoride out, you'd have to make the potassium come out too, and we do that by the obvious way. Just dump in valinomycin of potassium ionophore, which we also refer to as vitamin V. And then what will happen is that those guys are going to dump out their fluoride. And notice how the blue got a little bluer on the outside. We have a fluoride-sensitive electrode here, some electronics. And so I'll show you I'll fabricate some data, which I like to do often. Oh, and we can add detergent and get a sort of a normalization value for all the fluoride there. So you might get some recording that looks like this, valinomycin and then detergent. And the key, of course, to know what the unitary transport rate is the rate, and that is okay, so we're looking at a difference in accent or dialect here between Ireland and Boston. In my PowerPoint, that thing, that double arrow, is a tau, a time constant. And that's how we get the rate. And so now I'm going to show you some actual data. So, and I should say, this is a highly sophisticated instrument. And I thank God I'm a Howard Hughes investigator and can afford it. It's a beaker with a stir bar in it. That's basically it. And here's what the experiment looks like. We add valinomycin. Boom, the fluoride comes out. Add detergent, comes out some more, which shows us that we're in this unitary region. And in fact, what is that rate? Well, that rate is faster than we can measure because it is rate limited by the response of the electrode, which means that whatever the rate is, it's greater than 30,000 per second, which isn't a million, but it's greater than 30,000 per second, but it isn't 100. So it's kind of implies this may be a channel. And if it is a channel, it's an extremely selective one, because if we do exactly the same experiment with chloride and a chloride electrode, the thing just doesn't come out at all. So already we have an interesting case here. Whatever this thing is, it's very selective for the smaller fluoride ion over the larger chloride ion, at least 1,000-fold, and I'm going to show you later it's at least 100,000-fold, which is a very, very selective channel, as I will show you. So yes, it's a channel, and to do that, of course, we're going to do electrophysiology. We do some things and put them into planar bilayers. And I'll just show you a few channel recordings, single molecule events, single molecule insertions into the bilayer. Right here is the first insertion, holding at a negative potential, so opening is down. And what you see is that this is one of those single channels. I'll show you another. Here's some that have 3 channels happen to pop into the bilayer. But in these recordings, what you see is the channel is almost all the time open, just hangs there, sort of stupidly open, letting fluoride go through. Every once in a while it closes. Every once in a while you see a little half state. But basically it's 95+% of the time open. It does not gate. It's not voltage dependent. It's not pH dependent. It's not regulated. It's just pretty stupid thing, except it's very selective. And if you think about the physiology in a microorganism, it had better be selective. Because if chloride could go through that thing, this would seriously, remember, in a bug, chloride is about 1,000-fold higher in concentration than fluoride is. And so it would depolarize that bug, de-energize it. It would make serious problems. So actually, the selectivity makes some physiological sense, even though it doesn't gate and it won't be a plaything for gating channel. People who like channel gating won't like this guy. Anyway, so this is over the single channel current says you've got something like 6 million ions per second going through there, and that's not a transporter. That's a channel. So enough of that. I'll call it a channel from now on. So how's it built? And we're going to really now go into just talking about what does this thing look like? How's it built? And it is built weirdly again. So fluoride itself is weird. The physiology is weird. And the molecular architecture of this thing is also weird. And the weirdness is it's built as a homodimer. We imagine a pore down through a center of, you saw there was looked like a single pore there, pore down through the center of a channel. We don't have any examples of channels with a single pore that are made. Well, as you know, channels are built mostly by a barrel stave architecture, multiple subunits around a central axis. We don't have any other examples of a two-stave barrel. But this would be, we imagined at the beginning here that this was a two-stage barrel. And don't look at that, it just shows that there's some data in back of that, and there's a lot of other data in back of it, and you'll just have to see what an honest face I have, and believe me, it's a homodimer. But it's a strange homodimer, because here's the conventional way of putting two subunits together, or say 4 subunits. If we were looking at a potassium channel cartoon, we would see a blocker coming in there, tetraethyl McKinnon blocking the potassium channel. But in fact, that's not how this guy is built. This guy is built this way. It's an antiparallel homodimer, or that's called dual-topology homodimer, where the 2 subunits are literally in opposite transmembrane orientations. And again, I'm not going to show you all the boring cross-linking and all that kind of biochemical stuff to show this, to try to convince you. I'm just going to go and show you one set of experiments I'm extremely fond of that I think nailed it before we had a structure. And that relies on a series of proteins called monobodies. These are engineered, designed proteins that are selected by our collaborators, Shohei Koide and his lab, from phage display libraries. They're based on a little protein domain called fibronectin 10 domain, where Shohei has developed libraries of 10 to the 10th, 10 to the 11th copies of randomly varied or diversified sequences in certain parts of these things in hopes of making them like sort of antibodies to bind to a target protein. And we've selected out of this library 8 such sequences of monobodies that bind with nanomolar-ish affinity to our homologs, our Fluc homologs. And of course, we wanted to use these for crystallization chaperones. That's what you would want to do this for. And I'll show you that later, but in fact, they had another property which was extremely useful for us to show that these really are antiparallel homodimers, and that is that these monobodies in binding also block the channel. So here's what, again, a channel record looks like at a slow time scale. Notice, almost all the time open, occasional full closings, occasional halfway closings, but basically the thing is open all the time. You now add some monobody and you get a recording that looks like this. A different monobody will give a similar looking, but with different kinetics and different KD recording. And I think what you all realize is what you're seeing here is at the single molecule level, single channel being blocked by a single monobody comes on, shuts it down, dissociates, the next one comes on, dissociates, and so on and so forth. And so these are blockers. Now, so what? So what? We can use this to design a very simple experiment to show that these have to be antiparallel. And the experimental design is this. It's very simple. You take a single channel, you put it in a bilayer, you add the monobody, and it's going to bind to some epitope there. I don't know where the epitope is, but presumably it's somewhere around the pore, and when it binds there, it plugs it up. But of course, this is a dimer, so it has to have a second epitope, and that epitope has to be on the other side. So the experiment is obvious. You put it on the one side, you see if it blocks, you now wash it away, better stop blocking. You now put it on the other side. Duh, if it blocks, well, that says the epitopes have to be on both sides, and that means it's got to be like this, I think. I can't think of another explanation, and of course, if it wasn't that way, I wouldn't have showed you that slide, but it really worked beautifully right the first time. Put the monobody on the, whatever you call it. You can call it the outside, although it's equivalent, if you think about it, to the inside. And then, you wash it off, and there it is. There's the channel after the washout. Now you put it on the other side. So this double-sided perfusion experiment, I think, just nails it. It says the epitopes are exposed on both sides, and therefore the things are antiparallel. And just always liked that experiment. Now, I have to remind you all, many of you will realize this has been seen before throughout the last bolus of amino acid transporter structures that have been coming in at us over the last 10 years or so. It's the Lu-T fold is a great example of that in this first amino acid transporter from Eric Guo's lab, is that they have this inverted topology within the polypeptide, so that the transporter has one side. If you look at the structure, this part of it is upside down with respect to that, even though you don't see that in the sequence at all. But once you see the structure, you see that this part intertwines with that part, and if you turn it upside down, it lies right on top of it. So this is a prevalent thing that we see out there in the structural biology of membrane transport proteins. And this probably is something that has evolved from a duplication and then an inversion of two things, just like the fluke, but then got locked into the same polypeptide. Anyway, that's just kind of an interesting aside. Of course, the next step is to get the structure, and I'm just going to go right to the chase. That's what it looks like. We had to have monobodies in order to make the crystals. That would not form crystals without monobodies in complex with it. It's made of this is the polypeptide, the single monomer, has 4 TMs, and the third TM right here is broken in the middle. Right in the middle, it looks almost like a transporter in that regard, and that red spot is the most conserved part of the Fluc proteins, which makes us think, oh, the pore must be there, the selectivity filter must be around that part, and if we could only see the structure. Now, I want to see, how do I make this thing go? I need some help back there. You made it go before, let's see. No. Is there a way to make the movie run, or are we lost? Okay, I think we're lost. All right, folks, I'm afraid I can't show you a movie of this thing, but you'll see some other static structures later on, okay? Let's just go on. There's something not quite right about the structure. And this is, we got this structure, we saw it, we saw where these conserved places cross each other, and right in the middle. The thing looks kind of like an hourglass, but there's a problem in that we couldn't see a pore there. These are the aqueous vestibules. If you saw that movie, I would have shown you the monobodies sitting on top and the bottom plugging those vestibules. But these are the aqueous vestibules, and there's the crossover, that red part, that very conserved part. And you can see from a slice down the middle, it looks like what a channel should look like. Has 2 vestibules on the side and a narrow place in the center, except we couldn't see any pore there. It looked like it was closed. It was just a big plug of protein. But our electrophysiology says it's almost never closed. So what's going on here? It was a worry. And more, you might say, well, just see where are the fluoride ions? And there's a bit of a problem. These were rather low resolution structures, 3.5 angstroms, where you could not hope to see fluoride. And even worse, the crystals only formed in the absence of fluoride. So go figure, they always stomp on you somehow. So that was just the time when I was heading off to Oxford for a year, and gave me some time to think about what could be going on. And luckily, I hooked up with Simon Newstead, a young crystallographer in the biochemistry department there, who had some expertise that I didn't have and knew how to make certain kinds of crystals that I didn't know how to make. And we spent a wonderful year playing crystallography together and using the wonderful diamond synchrotron. And at the end of that year, we had 5 crystals forms with 2 different homologs and 5 monobodies. But the really important, and these all lined up in the channel part, although the monobody parts were different monobodies, so they had slightly different things, but they all stuck their heads into the vestibule. And the important thing is that both of these were fairly high resolution, and they were in the presence of fluoride. So now we can look and see what's going on. Where's the fluoride ions in there? So that's what I'm going to show you. There were, when we look at these structures, there were 5 mysterious electron densities that were nice and round and looked like ions. And so I'm going to just tell you what these are. The one in the middle, where the pore we thought should be, is a sodium ion. And it is a sodium ion that is simply irreversibly buried in this thing, and as probably, as I would speculate, as a structural element in the dimerization that helps hold the dimer together. It never comes out once it's in. It's completely buried, but the other 4 blobs are fluoride ions, and here's where we find them. 1, 2, and then copied in an upside down way, 1, 2, in this sort of symmetric way. So it looks kind of as there are 2 tracks along the sides of the hourglass, not in the middle, in which the fluoride ions live. And if we look closely now at what's holding these fluoride ions in there, we see some other weirdness, a very strange thing, which we're calling the phenylalanine box. There are these 4 phenylalanine residues. Those are absolutely conserved phenylalanines, I should say, in that region. And they are actually coordinating or helping to coordinate the fluoride ions. So if we look at one of these tracks, this one on the right side, closer up, what we will see is that the fluoride ions are coordinated by some hydrogen bond donors, as you might expect, because these, remember, fluoride loves protons. And so there are serines, there are asparagines, there are histidines that are in the coordination shell, but in each shell there is 1 phenylalanine. Now you might think a phenylalanine residue should not interact with an anion. That's crazy. It's just a hydrophobic weird thing. But it's not weird. It's not crazy, because remember about aromatics. They have electron clouds sitting on top and on the bottom of the ring, but that means, and there's a little excess negative charge, but that means there's got to be an excess positive charge on the ring itself. And in fact, that's true. The hydrogens of the ring, of aromatic rings, carry a little excess positive charge that balances the pi electrons. So if you have an edge-on approach, that makes electrostatic sense. This is something that has never been seen in the protein database before, but it has been seen in the small molecule database of halide ions being coordinated by the edges of aromatic rings. And we think that the hypothesis is that this is something that makes this thing special for fluoride. It has something to do essentially with the fluoride binding sites and maybe the fluoride selectivity. And so I'm going to finish up with a few experiments that are just now concluding to go sort of a little bit after the role of those phenylalanines in fluoride binding and selectivity. Of course, and that's the electrostatics of a benzene molecule. There is a little blue on the edges to match the red on the top and the bottom. Of course, the speaker always says he's about to finish up, but he knows he's got another 75 slides, but he has to give the audience some hope. So here it is. So what I'm sort of saying here is that this looks like it's a double-barreled channel, that there are actually two pores that sort of start and end in those vestibules, so that when a monobody would stick its head into there, it would block both pores simultaneously, which is sort of what we see. And so now I'm going to just look at more closely, are there really, so here's the problem. I came late in life to crystallography, and I distrust it in some ways. I like it, I've become addicted to it, but I do distrust it. And especially, I distrust dry crystallography. Crystallography where all you see is a structure, and then you start psychoanalyzing the protein function. And so I would like to see some sort of functional manifestation. If I'm saying there are two pores here, I would like to see something functional that supports that before I put too much emotional energy into saying, it's double-barreled. So that's what this is about, to try to give some functional vitality to this otherwise dry crystallographic conclusion of two distinct fluoride transport pathways. And what we're going to do is look at these phenylalanines and mutate them and see what happens, and see if we can somehow manipulate individual pores. Those are just my nomenclature for this. So I'm going to go back and use that all or none flux assay in liposomes that says, is channel there or is channel not there? Highly quantitative flux assay. So we're going to take the wild type, and we're going to say, well, let's make a mutant and crystallize that mutant and look at the fluorides in there. And here's what we see. So we're going to take one of those phenylalanines, this one, mutate it to isoleucine, crystallize that, and look at where the fluoride densities are. And what we see is, that density has disappeared. But only that density, that one that is still coordinated by the phenylalanine that's left, is still there, nice and robust. And if we do the same thing with the other one, that fluoride remains, that fluoride does not. And these mutants are superimposable in the crystal. Well, what about the function? What about the function? We'll go and we'll look at the highly quantitative channel, no channel assay. That one, there it is. That one is happy, it has a channel there. That one, those two are dead. There's absolutely no fluoride transport there at all. Okay, so what else can we do? The problem now is, can we use this to isolate one pore or the other? And the way we would like to do that, it's a homodimer. So if we could link those together and now mutate only one side or the other, that's how you could go after this, right? But of course, to link them together, because they have an even number of membrane crossings and one of them's upside down, the linker has to have a transmembrane sequence in it. So we built an artificial tandem using a foreign transmembrane sequence, just an old greasy thing, and lo and behold, the thing expressed, very lucky, and it had all the properties we would expect of the wild type channel. So this tandem was a good, it didn't express enough to crystallize, but expressed enough to reconstitute and look at its function. So this now puts us in position to do the experiment we really like to do, which is to kill just one side or the other and see what happens. So here we go now in these tandems. If we kill just that phenylalanine, what do we have? Happy channel. There's a channel there, even though we've knocked out one of the phenylalanines. Let's make a double mutant. Make a double mutant there. That double mutant would just mimic the mutant that we made on the homodimer. And of course, that's dead because we already showed that's dead in the homodimer. But what about that double mutant? That double mutant's dead too. What about that double mutant? It's perfectly alive. So I think that's just a qualitative indicator that, hey, there really are two barrels here. Realize these mutations would have been uninterpretable without a structure. I mean, just imagine we make phenylalanine mutations, and sometimes it's alive and sometimes it's dead. We didn't know the structure. These would have been senseless. But with the structure, they actually make some sense because they're predicted to be this way under the hypothesis of 2 pores. So what we'd really like to do now is look at single channel recordings from 1 pore channels and see what do they look like. And I'll just show you that. So here's the wild type channel. It looks exactly the same as the homodimer. It's got a conductance of about 10 something like 10 picosiemens. And then we take that mutant, and I should say, I'm just going to show you one of them, but we've done all of the combinations of the single pore mutants. And what you get is that. Oh, and I wanted to say one other thing before I show you that. I think some of you have noticed that there are these rare half closings. And Lucia, I know what you're thinking. You're thinking those are rare fluctuations of one of those pores to be closed. That is, one of those pores completely blocks itself in some weird fluctuation so that now the other pore is there, okay? So that would predict that if we would now look at the single channel of this guy, we should predict 2 things. One is the channel should be, if everything is simple, about half the conductance, and those things should go away. Those shouldn't be there. Half closings shouldn't be there if those represent a single pore. If you've only got a single pore, they're going to close all the way. But that's not what we see. We do see that it's half the conductance, but those little guys are still there. So we don't know what that is, but it's something that has to, I think it's some fluctuation that acts in the vestibule, which is an electropositive vestibule that somehow is modulating the access of ions to both of those pores. But, you know, go figure. I have no idea what that is. So I think that sort of finishes to the point where we are now. We don't really know why chloride never gets in there. I have my ideas, but they're just sort of, you know, out there floating around, floating free in outer space, as they say. But I think one of the questions one would want to ask right away about selectivity in a place like this, especially in light of the fact that our substrate is a fluoride anion, is why would fluoride ever want to be in a region like this? There's almost no water. It's a very narrow crevice. And fluoride in aqueous solution, you should appreciate, is the most hydrated monovalent ion in the universe in aqueous solution. And so the energy of taking all that water off of the fluoride and taking a naked, unhydrated fluoride ion and sticking it into the middle of a protein, that energy, if uncompensated by the protein in some way, would be prohibitively expensive. And fluoride would never see this as an anion-happy site. Well, now we know that I think we know that the mutations of that phenylalanine have told us, or have suggested pretty strongly to me, that that phenylalanine interaction is important for making that site happy for fluoride. And I don't quite know how or why, but I think that's going to be a critical aspect of selectivity, that you have this funny, weird phenylalanine halide interaction that makes the fluoride be perfectly happy to be in this place without its water shell, and not so happy that it binds so strongly that it never comes out, because remember, 6 million ions per second have to go through there, which means 6 million times per second this thing dissociates. So I don't know the answer to these things, but these are the kinds of chemical questions that I'd like to go into in the future. And so I think I'll just finish up there, urging you young physiologists again to think about weird weird places to find strange physiologies. There's a lot of this is very new field. There are only 10 papers in the field. You know, it's easy to keep up on the literature. And 5 of them are mine, so, you know, I can send you them. I'd just like to blame the guilty parties here. You know, I don't do a lot of these experiments myself. I do a few, but mainly I'm sitting in my office answering emails all day. Randy Stockbridge was a postdoc who really brought this, who started this project in the lab. I'll just tell you, when I originally heard from Ron Breaker about these, I said, oh, Randy had just arrived in the lab as a new postdoc. I said, this is great, we can work on CLC fluoride. And because we know a lot about CLCs, and this is a perfect project for you. She said, no, I'm gonna work on that other weird thing. And I said, no, you're not allowed to do that. But of course, she did that anyway, because that's what they always do. And I say, no, it's good. Reverse psychology works. And on 3-year-olds and postdocs. And she's done she started the project. She worked out all the biochemistry. She did most of the original crystallography. And she's now in her own lab at University of Michigan looking for postdocs of her own to torture. And Nick Last is a postdoc who's recently come into the project to do these single-pore tandem experiments. And you can see he is not trained as an electrophysiologist. He comes with a chemist background, which means I cannot convince him that you don't really need gloves for single-channel recording. But he's worried about those high currents of 1 picoamp that might electrocute him. He's a little nervous about that. And I have to acknowledge my wonderful collaborators, the Coitas, without whom we would not have had the monobodies and therefore would not have had the structure and would not have had the experiments that show these are upside-down dimers. And Simon Newstead, who saved our butts during my sabbatical at Oxford. Thank you.